We are now ready to begin part 3 of chapter 13, and part 3 deals with mutations, when things go wrong with either the transcription or the translation process. So by definition, a mutation is a change in the DNA that affects your genetic information. So if you look on the internet, you can see all sorts of horrific examples of mutations. On this screen you can see the two-headed snake up in the corner, and then you can see Raphael, who was just a regular turtle until they was transformed by a mutagen in his environment and became a teenage mutant ninja turtle. We're going to be looking at several different types of mutations here. This picture gives you sort of a brief overview that we started with our original chromosome, and through one of 4 different processes have ended up with something completely different, and we will talk about each of these in more detail. We are going to differentiate between our mutations and break them into 2 general types. Type 1 we're going to call just a gene mutation, and with a gene mutation we're changing just a gene. It doesn't mean that it's not going to affect the organism, because it certainly could, but it's only affecting one gene. The second type of mutation is chromosomal. We generally, this will be the lack of a chromosome, so not enough chromosomes, or too many chromosomes. And then we can also look at other types of mutations where parts of a chromosome break off or get omitted or become inverted or switch places. So we're going to begin by looking at the simplest of mutations, which we call a point mutation. And basically this says that at some point on the molecule, a nucleotide has been changed, or a couple of nucleotides have been changed. So it's just a change at one point. You could have multiple point mutations, but we're not going to see an entire part of a chromosome gone. It's just going to be something in the sequence. We're just missing one of the bases, or have too many bases, or they've switched places. So what we're going to look at now are the types of point mutations. In the next few pages we'll see what a substitution mutation looks like, an insertion mutation, and a deletion mutation. Okay, our first type of point mutation, we're going to look at substitution. So if you take a look at the picture, we begin with our DNA strand and our mRNA strand, and these are the amino acids that we would end up with. But when substitution takes place, the C that was in our DNA has been substituted for a T. And the mRNA codes just like it's supposed to, thinking that that's a T, so it'll put in an A, and you can see that we get a completely different amino acid in that. So generally this affects a single amino acid, and it's usually caused by changing one base. And this will be true of most of the mutations that we talk about. It could be completely harmless. You could have this mutation in your body and have no idea, or it could be lethal. It could be something that is deadly, and when this mutation occurs, the organism ceases to develop. And it really depends on what's changed, what part of the code changed, what amino acid was changing, and it's kind of a crapshoot. You don't know what's going to happen. Here's just another look at substitution. So if our original strand said the fat cat ate the rat, with substitution the C in cat was changed to an H, then we have the fat hat ate the rat. Close, but it really does not mean the same thing. An example in humans that is a point mutation is sickle cell anemia. This picture that you see here are normal red blood cells, and they sort of look a little bit like cherry lifesavers. They're round, and they have a dent in the middle. Well, with sickle cells, they are sickle-shaped, which is kind of a half moon. Well, half moons, you can see in picture number 1, tend to get stuck as they're going through the arteries, and they start to bunch up, and they can form a clot, and this can result in a very painful episode and can also lead to death if the blockage is complete. So that was substitution. Insertions and deletions are oftentimes create something that's much more visible or create a change that is more dramatic or more lethal. And we call these frame shift mutations. If you think about watching a movie, and a movie is made up of these individual frames or a comic strip, if you shifted those, the movie wouldn't make sense, or the comic strip would no longer make sense. And that's what happens with these frame mutations. So if you want to write the biology definition, it shifts the reading frame of the message. And because of this shift, the protein might not do what it was intended to do. Just like you would be confused in a movie, if the frame suddenly shifted, your body gets confused, this protein becomes confused, and it doesn't know what to do. So let's look at insertion, an insertion mutation. And this happens not when we switch, when we substitute, but when we actually stick an extra base into the sequence. So when you look at the top, our DNA strand was TAC, and with insertion, we inserted a T, and it shifted everything down. So you can see now that every single codon in that DNA strand is different, and we have this lone T hanging out here at the end. And if you examine the messenger RNA strands, you can see that they are completely different. And as a result, number 1, we don't have a start codon, so we probably wouldn't even make this protein. And you can see that the next 2 amino acids are completely different. So using our same sentence, the fat cat ate the rat, with insertion, the fat cat ate the something, the fat cat confusion ate the rat, so we really don't know what that means. That sentence makes no sense. Here's nothing for you to write, but just take a look at, here's 4 different diseases in humans that are caused by insertion. And as you examine this picture, you can see with each of these, the codon that is inserted completely throws off the protein that is being made. I think one of you picked one of these diseases when we were talking about genetic diseases. Here's Huntington's disease, a deadly disease. I want to say that one of you did Fragile X, but it just shows that insertion can be something that is very horrible and may be life-ending. Okay, and the third type is deletion, where we have replaced a base. We've just taken it out. In the picture here, we've removed the H from the word the. It's gone. And as a result, our DNA strand shifts. So we have that frame shift going on, and the sentence no longer makes sense. And there's another way of looking at it. From the fat cat ate the rat, the fat ate the rat. So we're not, if you're reading that, you would be confused. You wouldn't know what happened. We're going to take a look at chromosomal mutations. Those were the mutations we just talked about, were genes, changes in a single gene. Now we're looking at changing the chromosome itself, the entire chromosome, either adding, removing, or changing the structure of the actual chromosome. These also come in many forms: deletions, duplications, inversions, and translocations, and we will take a look at each one of those in the slides that follow. A new word, polyploidy, having extra chromosomes. Very, very common in plants and in animals, even in us. Some genetic diseases and disorders are caused by polyploidy, and I have a couple pictures to show you on the following. So here's an example of polyploidy in strawberries. You've probably had strawberries like this, where it seems like two or more strawberries are growing together, and that is actually a genetic mutation. So you've eaten mutant strawberries. Here is an example where there's an extra chromosome in every single set. So in our 23, which should be pairs, we now have 23 triplets. And as you read below, and this is nothing that you need to write down in your notes, these babies who have the triploid syndrome usually do not survive. Another example of a chromosome mutation is Down syndrome, often referred to as trisomy 21. And what happens are these children are born with 3 copies of their 21st chromosome. Another picture of a very little baby with Down syndrome. You can see some of the characteristics, kind of the sunken in face, the flattened nose, the different eyes, the kind of a pronounced face, and it does generally result in some sort of intelligence issues and learning issues. Another chromosome mutation is the Creux-Duchat syndrome. We've deleted material on the 5th chromosome, and again, nothing that you need to write or know, just some examples. And again, usually results in various levels of mental handicaps. And the babies who suffer from this syndrome have a cry that sounds a lot like a cat, like a meow. Okay, let's look at a deletion. So we've lost a part of the chromosome, so we're not looking at just bases here. When we say A, B, C, D, E, F, we are talking about actual chunks of the chromosome. And you can see with deletion, we've lost part of it. Our B has been deleted. And then our circle here is our chromatid that would have held our 2 chromosomes together. With duplication, for some reason, part of the chromosome repeats itself. So our original, again, you can see the mutation is occurring here in the B section. Now we have 2 Bs. Completely different chromosome. With an inversion, different parts of the chromosome get flipped. So with this one, our DE flipped, became ED, and went to the front, and the BC flipped and went to the end of the chromosome. They became inverted. And then translocation. One part of the chromosome breaks, and then it attaches to another chromosome. So we had our ABCDEF. The DEF broke off, so that was removed. It was replaced by JKL, and that DEF hooked in to where JKL used to be. You can see that this is changing multiple chromosomes, and as a result could result in even worse changes for the organism. Alright, so now that you're scared to death and wondering how in the world do good things ever happen, most mutations don't do anything terrible. Most of them are small, happen. For example, eye color. When you see animals or people who have 2 different colored eyes, that is a mutation. Birthmarks are an example of a mutation. Most of us have them. Most of us do not have them in the exact same spot as our parents. It's just something that changed. Some of these mutations are harmful, and some we've talked about. So sickle cell anemia is a harmful mutation. Down syndrome would be a harmful mutation. And sometimes mutations are beneficial, and we want them, and after a mutation occurs, to try to keep making that. So one of the benefits to sickle cell anemia is people who did have sickle cell were immune to malaria. Sickle cell is more common in African Americans, and that also happened to be the area in Africa where malaria was very prevalent and very deadly. So sickle cell prevented those people from getting malaria. We're starting to see mutations now that provide an immunity to HIV. So what in the world causes these mutations? Basically, it comes down to two ways. Way number one is they can be inherited. So if your parents had sickle cell anemia, the chances of you having it would be greater. But mutations can also be acquired, and we do a lot of things to our bodies that could cause mutations to occur. Environmental damage. Number one would be exposure to the sun. Number two would be smoking. So, and number three would be a diet. So, believe it or not, what you eat can cause changes in your DNA. And then simply there can just be mistakes that happen when the DNA is copied. You have so much DNA in your body, mistakes are bound to occur. But luckily, most of them are not harmful, and we don't even know they happen. So again, I have a few questions for you to take a look at. Put these in your notes, and we will go over them in class tomorrow. Number one, a mutation in which all or part of a chromosome is lost. Choose which of those you think it is. Number two, a mutation that affects every amino acid following an insertion or deletion is called what? Number three, a mutation in which a segment of a chromosome is repeated is called. Number four, the type of point mutation that usually affects only a single amino acid. Number five, when two different chromosomes exchange some of their material, the mutation is called this.